beginning, and you go through maybe 10 to 12 years of process to develop this drug. Thank you. Okay. Last time we were talking about the clinical development plan, and we're discussing this draft Gantt chart. And the main thing is about the entire drug development process. Like I said, it may take 10 to 12 years, starting from the we call it SOD, start of development, sort of after the compound left the discovery lab, and they take quite a bit of time to get a drug developed into a successful drug, to get the we can submit this NDA, new drug application, and eventually, hopefully, the drug is good, efficacious, and safe, and FDA can approve it. So we talked about phase 1, phase 2, phase 2A, phase 2B, and phase 3. Now, I want to spend some time to talk about single rising dose and multiple rising dose. All right. Some companies call it SAD, single ascending dose, or MAD, multiple ascending dose. And in our company, we call it single rising dose or multiple rising dose. The idea is that you expose the test drug to a group of subjects, and this time they're healthy, normal subjects. And if you don't observe any adverse events or any problems or any safety concerns, you recruit another group exposed to a higher dose. And then if it's safe, you expose another group with even higher dose. So the doses are escalating. So these are what we call dose escalation studies. So start very low dose, if it's safe, you escalate, you escalate, you escalate, you escalate. And the single rising dose is that every subject takes just one dose, just once, okay? The confusion part about multiple rising dose is that it's the same dose, but multiple times. So in other words, the same dose strains. So again, this is you expose a group of subjects with the lowest dose, and it's safe. And now this time you expose the subjects to multiple doses, so more than once, but the same dosage, the same strength, very low dose. Now it's still safe. Then you escalate to a higher dose with a different cohort of patients or subjects, and then you keep escalating. So these are the phase 1 SRD and MRD, single rising dose, multiple rising dose. And then you get into phase 2. Phase 2, the typical phase 2 study, the first phase 2 is what we call POC, proof of concept. And then the next phase 2 would be dose ranging. And then you get into phase 3. If these are all successful, you get into phase 3. And phase 3 are typically long-term, large-scale trials to confirm the efficacy and safety of this drug. If phase 3 is successful, then you file an NDA, new drug application, to FDA, and hopefully the drug can be approved, and that'll be a successful drug. So that's the sort of a grand scheme or a first view of this drug development process. You start from preclinical discovery or research, you find the new drug, and you go through the preclinical development. And in the preclinical development, including the toxicology studies, formulation studies, animal PK, and all these kind of studies. And then you eventually get into human. Once you get into human, that's clinical trial. And you have clinical trial phase 1, phase 2, and phase 3. After the drug is approved and available on the market, you engage in phase 4. So last time we talked about the clinical development plan is actually a blueprint for drug development, which describes the draft package insert, which is your draft label, is also known as TPP, target product profile. The timelines, as we can see, there's the timelines, resources and budget needed for registration filing. So once you can map out all the studies like the Gantt chart showed, you can estimate the resources you need to be invested and devoted into the drug development. And it integrates information from toxicology, clinical pharmacology, formulation development. So these are the preclinical data, all right? Toxicology, clinical pharmacology, formulation, animal PK, all these information, you have to incorporate it into the consideration while you're drafting that Gantt chart. And incorporates registration requirements. ICH, which is International Conference on Harmonization. FDA, which is the US drug regulatory agency. EMA, they used to call it EMEA, but now they call it European Medicinal Agency and other regulatory agencies, and evaluate results against a product profile. So these 2 points are important, all right? This point gives you where are the fundamental basic information you have before you design this CDP. And this is important because you want to know how the drug will be approved. What are the requirements from different regulatory agencies? How do you achieve that? So when you set up a plan, you want to look at where you came from and where you want to go to, all right? And then the CDP itself, clinical development plan, is a life document. It's not once you plan for it, it's set. No, as time goes on, you learn more. You learn more about the study drug. You may want to make modifications. You want to make improvement. You want to make changes. So it's a life document. That's your CDP, clinical development plan, and that was this kind of Gantt chart. All right. So now we start with the drug development process. We start with non-clinical, like I said, pharmacology, toxicology, animal PK, formulation, all these non-clinical tests. All these studies or experiments were conducted before the test drug goes into any human body. All right. If all these experiments are successful, there is an important step we call IND, investigation of the new drug. So what you do for IND is that you put all the information you learn from here together in a document, and then you submit it to FDA. Now, if within 30 days after FDA receiving your IND and there's no objection, you can get into human testing. So IND is a document sort of between preclinical and clinical, where NDA is a document between pre-marketing and post-marketing. All right, so this is an important document to get the drug into clinical testing, get the drug into human testing. All right, that's IND. Once you get IND and there's no objection from FDA, you move into clinical development. And clinical development, there's phase 1, phase 2, phase 3. And if they're all successful, you put together a new drug application, you submit to FDA for review and approval. And if the drug is successful, then you follow up in the post-marketing setting, that's phase 4 clinical studies. So that's a very high level view of the drug development process. And then we'll get into some other concerns or considerations, all right? So when we're looking at drug development, at every stage of the drug development, the 2 questions we're asking is, does the drug work? Is it safe? We're always asking these 2 questions. Keeping in mind that these 2 questions are almost impossible to answer, all right? Does it work? How do you assess, does it work? All right? So of course, when we design clinical trials, we look at primary comparisons, primary endpoints, and we study different doses and all these settings. But the fundamental point is that, does the drug work? So like we discussed in the first lecture, that there's never a way to objectively or scientifically say that the drug is efficacious. The best we can do is to say that the probability the drug does not work is controlled under alpha. So we came up with a good handle about drug efficacy. So we do double-blind randomized controlled clinical trials. We try to establish this kind of efficacy more from a statistical hypothesis testing point of view. Is it safe? Now, that's an impossible question to answer. all right? You can never say a drug is safe. Like I said, any drug, if it works, the only reason it works is because it changes your biological system. So anything changes your system, it has to be toxic. So you can never say the drug is safe. So what does it mean? What does it mean by the drug is safe? All right. Can you make a comparison to placebo like what you did in the efficacy? Of course not. I mean, how does it mean when your drug looks like a placebo? All right. I mean, the safety profile, there are different adverse events, lab abnormalities, and all that kind of stuff. There are all kinds of safety findings. There's no way you can say the drug is safe. I mean, anybody tested this drug would have some kind of reaction to it, and could be positive, but could be negative. Do you mean short-term safety, long-term safety? And these are, again, like I said, questions almost impossible to answer. Now, if that's the case, how can FDA approve a drug and claim it's safe? All right. Keep in mind, FDA do not claim the drug is safe. All right. Now, let me give you an example. Now, if a drug causes nausea and vomiting, would you say this drug is safe? Well, if you look at oncology, if you look at chemotherapy, almost all the chemotherapy would cause nausea and vomiting. So from oncology point of view, they say it's pretty safe, just nausea and vomiting, that's not a problem. But now if you're developing a drug for respiratory diseases, so patients already have difficulty breathing. And at this time, if the patient suffered from nausea and vomiting, that could kill the patient. All right, so it's not safe in a respiratory drug, but it's safe in the oncology drug. So you can never have a blanket statement to say the drug is safe. So how does FDA approve a drug? So here's what they say. For efficacy, FDA would say the probability the drug does not work is controlled under alpha. And then they say the benefit outweighs the risk. So that's how you sort of look at the drug safety. You're not saying the drug is safe, but you're saying the benefit outweigh risk. So it worth the risk. And of course, for our everyday decisions, we're taking risks. So it's a matter of describing the risks. So that's why you have the package insert, the drug label describing the adverse events, the safety experience, and all that. Well, in addition to efficacy and safety, there's other questions like quality of life and health economics, and you would hope that will help with your commercial development and stuff. So these are the additional questions. All right, the entire scientific process goes like this. You'll always start with a hypothesis, which is a question. All right. So you sort of propose a hypothesis, you propose a question, and then you try to answer this question. What you do, you design a study, you collect data, you perform analysis, and from the data analysis, you got some information, and hopefully you can digest the information and formulate some knowledge. Okay. Now, after that, you would have a newer question. All right. So I think statisticians are very familiar with the term of hypothesis testing. All right. Hypothesis testing is about answering a question. And there's another activity which we call hypothesis generating. Hypothesis generating is raising a question. So this process goes like this. You always start with a question, all right? And then you go through this process and you do the hypothesis testing, right? If you do the hypothesis testing and the testing says the drug does not work, you say, okay, we failed on this drug candidate, and we start with a new another candidate, a different drug or different way of developing it. All right. But if the drug works, then you would have a new question, right? You're generating a new hypothesis. So based on this new hypothesis, you go through this process again. All right. So this is a scientific learning and confirming kind of thinking process. Now, the thinking is from a scientific point of view, you sort of you're sort of accumulating information over time, from preclinical, you go through phase 1, phase 2, phase 3, you got the drug label, hopefully you got approval, you got approved, and you got drug label. So that's the scientific process. So as time moves from like today until maybe 8 or 10 or 12 years later, you hopefully will get a drug, you will get a drug label. So that's a scientific process. Now, today we're talking about CDP, clinical development plan. Planning is different. Planning is to look at backward, planning based on label. In other words, I talked about TPP, or target product profile, or draft label. I say that is very important, very critical. Why? Because at this stage, at preclinical stage, you need to think how you're going to label this drug. Okay, so that's your TPP. You look at this drug label. Because you want the drug to deliver these kind of information, all right, efficacy and safety or whatever you want to do, that's your best hope for that drug. Your dream product is your drug label. Now you have that on hand at this point in time. And then you say, in order to receive an approval with such a drug label, what should my phase 3 study deliver? All right, your phase 3 clinical trials will have to deliver the information to support the label, right? And that's how you design studies. You have to design your phase 3 studies to deliver the information, it'll support the label. Now, how are you going to design your phase 3 studies? So you need to have enough information from phase 2 to help you design phase 3. So in order to achieve drug label, you need to have this set of phase 3 studies. But in order to design these phase 3 studies, your phase 2 want to deliver this necessary information. So how are you going to design your phase 2? Your phase 1 has to give you the information to design phase 2. All right, so that's why the CDP, the planning process, is thinking backward from your target product profile, going back to your preclinical stage. So you have this drug candidate, you want to develop into a successful drug. So you're going to have a dream for this candidate, this drug candidate. Hopefully 8 or 10 or 12 years from now, you're going to have a very successful drug. But now you have to dream what that successful drug will look like. And that's your target product profile. And by looking at that TPP, the draft drug label, and then you can plan on your phase 3 studies, and you can plan on your phase 2 study, plan on your phase 1 studies. So that's how we came up with that Gantt chart. All right. So we're looking at the target product profile, where we want to arrive at, and then we think backward, and that's how we plan. So now let's get into the phase 1, phase 2, phase 3 clinical trials and the thinking behind it, right? Phase 1, we have to first differentiate between oncology drugs and non-oncology drugs, all right? The oncology drug development program and non-oncology drug development program are fundamentally different. Traditionally, the way you try to manage cancer is this. Of course, the first thing is to prevent. I mean, if you can do whatever you can prevent having a cancer, do it, all right? Prevention. Now, if you cannot prevent it, so cancer developed, then what you do, early diagnosis. The earlier you can diagnose, the earlier you can find out, the better chance you can survive from it, all right? So prevention, early diagnosis, and after diagnosis, so I know that I got cancer, all right? Then what? Treatment, all right? So the traditional way of treatment, of course, nowadays there are different ideas of treating cancer, but traditionally the way they do it is that the first thing is to remove your tumor, all right? So the tumor grows, you remove it. If you can surgically operate to cut the tumor off, to remove it, you do it. In many tumors, it's difficult to perform operations like brain tumor, throat cancer, and things like that. And then what you do radiation. So you have this radiation therapy. You try to sort of use radiation to kill these tumors. But then the thing is that either surgical removal or radiation, after that, we're concerned that we did not clean up all the tumor cells. Then what you do chemotherapy. So you follow chemotherapy. So chemotherapy is mainly trying to kill off those tumor cells. So if they can kill tumor cells, they can kill normal cells also. So most of the chemotherapy are known to be cytotoxic, being they're toxic to your cells, to your tissues, okay? So by nature of oncology treatment, the drugs are toxic to begin with. So that differentiates the cancer drug from the non-cancer drug, all right? So most of the chronic diseases like respiratory disease, autoimmune disease, cardiovascular diseases, many of these diseases, they are not life-threatening, but they can be chronic. I mean, patient may have this disease for a long time, like diabetes, high blood pressure, and all these kind of conditions. So in these kind of studies, your phase 1 start out with healthy volunteers. Okay. So let me backtrack a little bit. The phase 1, phase 2, phase 3 differentiation. Okay. Now, phase 1, the two most important deliverables of phase 1 are pharmacokinetics, which is what we call PK. Later, I'm going to describe what is a PK. All right. And MTD, maximum tolerated dose. Right? In other words, phase 1, you try to escalate the dose up to MTD. So beyond MTD, people may not be able to tolerate. Now, if you can establish MTD and PK, then you move to phase 2, and phase 2, you try to bring the dose down to minimum effective dose. And if you can successfully do these two phases, then you design your phase 3, try to confirm. You use large-scale, long-term trials to confirm the efficacy and safety of this drug. Now, in order to study PK, pharmacokinetics is more about metabolism. So how does your body work on the drug? We want to have a clean PK profile. We want to have a good understanding. Now, usually patients, their metabolism system may be compromised. So if you use patients in phase 1, then you may not be able to have a clean PK picture. All right. So typically for chronic diseases or infectious diseases, your phase 1 trial are healthy, normal volunteers, so they can get a clean PK. I described oncology because the nature of oncology drugs are different. So it's not ethical to use healthy volunteers in oncology trials. So in phase 1 oncology trials, you start with patients. So that's a different story, right? For most of the drugs, non-oncology drugs, you start with healthy normal volunteers, right? So phase 1, that's the first time the test drug is exposed to human. You always have a first person testing the first human dose, and often young, healthy individuals. And sample size is not very large, and trials usually not sized based on hypothesis test. Well, we'll talk about bioequivalent studies in a separate lecture, all right? And randomized placebo-controlled double-blind. Now, phase 1, we usually use a biomarker, all right? When I describe the Gantt chart, I talk about the beta blocker, all right? So because phase 1, you're using healthy volunteers, you cannot study their blood pressure because their blood pressure is normal. These are healthy, normal volunteers. So in phase 1, there's no way you can look at the blood pressure is going to be lowered because their blood pressure is normal to begin with. But at phase 1, what you can do is you can look at their beta agonist. So you can use beta agonist as a biomarker. In other words, if you're developing a beta blocker, And from your phase 1 trials, even though they're healthy volunteers, but their beta agonist is not changed, then you don't have to get into phase 2 because the fundamental mechanism of action of the drug, they did not achieve, all right? So you start with kind of biomarkers. So you don't get to see clinical endpoints because these are healthy volunteers. POCP being proof of clinical principle, all right? And later we'll talk about POC, proof concept. For oncology treatment, we start with patients. And for oncology, there's no placebo control. And there are always dose escalation in phase 1, all right? Either oncology or non-oncology dose escalation. And for oncology, they typically use 3+3 design, which I will talk about later. And continuous reassessment methodology or Bayesian logistic regression model. So these are mostly for the oncology phase 1 studies. All right. Now, in phase 1, like I said, for the non-oncology drugs, we sort of look at the pharmacokinetic property of your test drug. So we sort of assay drug and metabolites in biological fluids. So what is metabolite? So for chemical compound, the typical way the scientists design the drug is that they come up with a compound, all right? This chemical structure, hopefully this structure will help to reduce your beta agonist or trying to attack a certain kind of disease or something like that. Now, but this part of the chemical structure, it may not be stable, it may not have the desired property or something like that. So they sort of combine this with another part of the chemical structure. All right, so now it makes it stabilized, and then you can go through your GI system. You take it by mouth. But once you go through the GI system, your body will metabolize this drug. But metabolize is kind of breaking down this compound. All right, once it's broken down, it goes back to the structure you want the drug to be. All right, so that this structure will go to where the disease is, and hopefully that'll help the patients. So the broken down part is called metabolite. All right, so phase 1, we look at pharmacokinetics. You look at the drug or metabolites in biological fluids, mostly in blood, sometimes in urine. All right, try to understand pharmacokinetics and pharmacodynamics. So pharmacokinetics is what does your body do to the drug, and pharmacodynamics is what does the drug do to your body. And phase 1 trials are typically intense observation, usually hospitalized or hospital facility. So in other words, we will say these are sort of inpatient studies. So subjects go to the clinic or go to this facility, they'll stay there for 24 hours, 48 hours, 72 hours. So it's very intensive observation. The study nurse will observe these subjects, will dose these subjects and draw blood and take blood measurements and all that kind of stuff. So it's kind of inpatient setting. Identify and monitor target organ toxicity. Target organ toxicity based on toxicology results or other drugs in the same class. So at phase 1, you really want to watch safety. Remember, I was talking about dose escalation. Start with very low dose, you escalate. You want to be very careful. You don't want the drug to cause any problem. So you're looking at the drug safety. You don't want to see any toxicology or any target organ problems. So PK, PD, like I said, PK is how does your body work on the drug, and PD is how does the drug work on your body. Basically, you want to establish some kind of time profile. And this time profile will help you understand the frequency of dosing. So let's take a look at this picture. All right. Now, the blue line is the drug concentration in your body. So this is what we call a time concentration curve. All right. So before the subject is dosed at time zero, if you draw blood from here, from the subject, you're going to observe no drug level because the subject is not dosed. So you draw blood at here, you have 0 drug concentration. And now you dose the subject. And then after probably 15 minutes, half an hour, 1 hour, your designed time interval, you collect their blood sample, or you draw blood from the subjects. You collect the sample. And then you perform an analysis, see how much drug goes to the subject's blood. So it increases over time, right? And up to a point, it'll decrease. Okay. Then after 12 hours, you redose. So you measure this drug concentration in the healthy subjects' blood samples. All right. Now this yellow line is based on the preclinical studies. So from preclinical, you would establish some minimum effective level, drug level. You have to have this much drug in the blood based on the PD marker, right? Pharmacodynamic marker. Biomarker I just mentioned is probably like the level of beta agonist you're reducing or something like that. You're measuring that. And you want to maintain this level of drug concentration in the body. Now, for the blue line, so if you follow the subject's drug concentration, after 12 hours, it'll probably drop below the yellow line. So then the drug will lose its efficacy, all right, because your PD marker do not allow the activity you would hope the drug will deliver. So what you do, you redose, all right? So you redose, you have the newer blood concentration levels, all right? And then by 24 hours. So now this looks like a twice-a-day drug, right? So you study this drug, you look at the PK, you say, well, the time profile looks like it's a twice-a-day drug. And twice a day, we call it BID, all right? It's Greek word. QD means once a day. BID means twice a day. TID means 3 times a day. QID means 4 times a day. And there are all other ways of describing the drug frequency. All right. So, so here is your time profile. All right. Time concentration curve to give you this time profile. And now you see this time profile, and then you're in a team meeting, and then the marketing team member looked at the data and said, well, if you're developing a twice-a-day drug, there's no way I can market it because the competitor on the market is already once a day. Can you convert it to a once-a-day drug? And then the scientists discussed and said, well, maybe we can double the dose, all right? If you double the dose, the time concentration will look like this. And you can almost maintain a 24-hour dosing, right? So why don't we do this? You know, we double the dose, we get a once-a-day drug. So what's the problem with this picture here? All right. Now this point is what we call a Cmax, a maximum concentration. This is a Cmax for the blue line. This is a Cmax for the red line. Usually, these amount of drug concentration may trigger adverse events or side effects. So if this is the case, then you cannot go with double the dosing. All right. So that's where we say QD feasible, once a day feasible. If you can double the dose, and if high levels are well tolerated, okay. Otherwise, we need to default to twice a day BID dosing, or change the shape of the curve with control release. So when you look at drug commercials on the TV, whenever they say certain drug was dash CR, CR means control release. Control release will be a different formulation. All right, you reformulate. You have a different kind of tablet or different kind of capsule, something like that, so that the curve may look like this. All right. So the Cmax is not so high to trigger any adverse events, but you can maintain it for 24-hour dosing. All right. So these are some of the PK properties we're looking at. So you escalate dose, you hope to estimate the maximum tolerable dose. to establish a safety profile. And sometimes you keep escalating the dose, you still couldn't find any toxicity, you still have to stop somewhere, we call it maximum feasible dose. And you want to propose dosing regimen for phase 2. Now we got into phase 2. All right, phase 2 are patients with a target disease. All right, now because in phase 2 you're using patients, of course you can study efficacy. So it's the first time you can study efficacy. Sample size depend on your design. That's not necessarily small number or big number. In phase 2, you really want to evaluate whether the drug is efficacious or not. And you want to validate the efficacy measure, you want to assess safety, and you also want to explore this quality of life or health economic endpoints. All right, now phase 2. The first phase 2 is proof of concept. You want to be sure the drug is efficacious so that you can move on for further development. Okay, because before phase 2, we didn't get to see how the drug worked in patients with a target disease. So POC, proof of concept. All right, now from the proof of concept point of view, you try to look at the test drug differentiated from placebo. All right, if the test drug is efficacious, you say, concept is proven, then we move on. The next thing will be dose ranging. Remember, phase 2 is trying to lower the dose. We try to find the minimum effective dose. All right, so you start with MTD, or maximum tolerable dose you observe from phase 1. All right, you learned from phase 1. And then you say, I do a phase 2 design with MTD against placebo. So that's a POC, or proof of concept. If the concept is proven, I move on with dose ranging. So a typical dose ranging study look like placebo, low dose, medium dose, high dose. You have multiple doses, and you try to find out which dose is still efficacious, that's low enough. And nowadays, a lot of companies are combining both POC and dose ranging together. And then we have different statistical methods to deal with it. And then we'll get into the detail once we get into phase 2. And OLC being ordinal linear contrast test. MCP mod being another popular phase 2 method. And so once you combine POC and dose ranging, you increase the speed of development, but you're carrying more risks. Again, once we get into phase 2, we can be a little bit more specific about what I mean by these kind of trade-offs. All right. Now, this concept I will talk many times throughout my lectures, which is confirm and explore. All right. Remember, in the earlier part, we showed that picture about hypothesis, design, collect data, and perform analysis, information, and then you get knowledge and this scientific process. If you want to simplify it, it's just 2 steps. One step being exploration, the other step being confirmation. So exploration part is you raise a question, you're trying to understand what's going on. You try to explore, all right? Now, after you have enough exploration, you want to make sure what you learned is right. And at that stage, you want to confirm, you want to sort of check and be sure what I learned is correct or not. So that's the typical scientific process. You always start with exploration, and after you have sufficient exploration, you try to confirm. Now, how does statistics help with this scientific process? So we know there are 2 big things statistics does, all right? Estimation and hypothesis testing, all right? What is estimation? Estimation is about exploration. Right? You try to explore, you try to estimate, you try to learn. We do point estimate, interval estimate, we're learning, we're exploring. All right. Now confirm is hypothesis testing. All right. Explore is also hypothesis generating, asking questions. All right. After you see the data, you probably have more questions. It's hypothesis generating. But confirmatory part is hypothesis testing part. Okay. I want to go back to the first lecture about clinical trial. It's about separate the two distributions. That's hypothesis testing. You try to confirm the drug is efficacious. Of course, you can never show the drug is efficacious, but you can demonstrate the probability the drug does not work is controlled under alpha. That's confirmation. So this concept is absolutely critical important in drug development, because once you get into phase 2, this POC part is confirmation. And the dose ranging part is exploration. So in other words, in study design point of view, you want to confirm first, and then you explore. Phase 2, randomized controlled double blind. You try to establish dose-response relationship. You try to estimate minimum effective dose. You try to estimate dose range. The important part of this whole thinking is that with phase 1, you establish the upper limit of your dose range, and phase 2 establish your lower limit of dose range. Now you have a range of doses, you can move into phase 3. All right, you want to define the dose or range of doses for phase 3 studies. Once you come up with this dose range, you select one or few doses, you get into phase 3 for large-scale, long-term trials to confirm it. Sometimes you use active control in phase 2, but most times you don't need active control. So the big challenge in phase 2 is about studying these two curves, the efficacy curve and the toxicity curve. All right. So again, like I said, I'll get more detail of this picture once we get into phase 2 discussions. Now, those curves looks nice, but in reality, you never see these curves. The best case is you probably get to see at different doses, there are different responses. You try to guess what the curve look like. Well, phase 2, you have the powerful primary objective. And again, for study design, you have to be very clear about what your primary objective is and how do you achieve your primary objective. So you calculate study power based on the primary objective, and often underpowered for secondary objectives, underpowered for subset analysis for other reasons. Your secondary objectives are mostly exploratory in nature. They're not necessarily confirmatory, okay? And typically two-sided alpha 0.05, 0.1, 0.2 with pretty good power. So again, we'll get into a lot more details into phase 2. So if you're able to successfully establish the MTD, which is the maximum tolerable dose, the upper limit of your dose range, and you're able to establish the lower limit of dose range, which is minimum effective dose, then you move into phase 3. And of course, you have to demonstrate from phase 2 that the drug is both efficacious and safe. Now, phase 3 is a patient with target disease. So you want to mimic your phase 4 population. All right. So the design considerations in phase 2 and phase 3 are fundamentally different. All right. Because phase 2 you're exploratory, you're trying to show the drug works. So in your patient selection criteria, you want to be very, very selective, because you don't want, in phase 2, you don't want patient to be too sick, no drug will help them. And you don't want the patient's disease being too mild, because you cannot differentiate from placebo. So your phase 2 patient selection criteria are pretty tight. Phase 3 is different. In your phase 3 trials, you want your phase 3 patient population to mimic your market population. So you want to relax your inclusion and exclusion criteria. So patient with a targeted disease and this less homogeneous patient population than your phase 2 studies, by relaxing your inclusion and exclusion criteria to match the phase 4 population. Traditionally, FDA require a replicated evidence. So you want to have 2 or more trials with hundreds of thousands of patients. All right. Now, phase 3, you're committed. All right. You say the drug is good. We want to move to phase 3. So it's a huge investment into phase 3, long-term, large scale. All right. Hundreds to thousands of patients. You want to have at least 2 trials so they can provide replicate evidence. You want to confirm efficacy, and you want to demonstrate long-term safety. Right? Sometimes you have subset analysis. All right, and in many cases, phase 3 are comparing with active control. There's already a drug is approved on the market. You want to compare your test drug with the active control. You want to establish safety in large number of patients, patients with concomitant medications and comorbid conditions. And phase 3 trials are pivotal trials in registration dossier. So these are the trials go to NDA. You want FDA to look at these trials and they can approve the drug. Randomized controlled double blind and powerful efficacy. Sometimes you power for safety, that's a different setting, right? And you perform subset analysis. So that's your phase 3 setup. Phase 3 typically two-sided alpha 0.05, greater than or equal to 90% power. All right. Now, in clinical trials, other than bioequivalence and biosimilarity, the clinical trials are one-sided in nature. All right, because if your test drug is worse than placebo, you're not going to develop it. It's one-sided. So that means one-sided alpha 0.025, greater than or equal to 90% power. Sometimes you include interim analysis, and of course, in certain situations, you have data monitoring committee to watch your phase 3 trials. And phase 3, sometimes you get into non-inferiority trials, and phase 3 is important to look at quality of life and health economics data. Now, after phase 3 is successful, the drug will be approved. All right, so phase 3 are for registration purposes, confirmatory hypothesis testing, and you want to study for target dose or doses. If the drug is approved, you get into phase 4. Phase 4 study is for large-scale safety surveillance or new indication. And sometimes after the drug is approved, you may have to change the label dose post-marketing. And of course, many times there's post-approval commitments. So FDA will say, well, approve the drug, but you have to commit after the drug is approved on the market, you'll have to do more safety studies and want to see more data. Registration. The registration is a process. So we start with pre-NDA meeting. Of course, we have different names. In US, we call it new drug application. In Canada, we call it new drug submission. In Europe, we call it market authorization application. Japan, NDA. And the pre-NDA meeting is really trying to look at whether the submission is acceptable or not. I mean, your pre-NDA meeting, and then you put the package together, and then FDA will do a preliminary check to see if you have all the necessary components. Now, if it's unmet medical need, you may involve the accelerated review or something like that. And sometimes you may get into advisory committee meeting, and there will be label negotiation, and there usually for FDA there's a 4-month safety update. That's after your NDA, 4 months after NDA, you do a safety update. So in general, phase 1, phase 2 are exploratory in nature. All right. Phase 1 says how often. You look at dose frequency, PK profile kind of stuff. Phase 2 is how much. What is your dose or your range of doses? And phase 3 is confirmatory. Phase 3 drug can be approved or not. And then if it's successful, you go to NDA, and phase 4 are more scientific, commercial. And we want to know the regulatory requirements. All right. So we want to have the regulatory knowledge and interaction is essential for successful and efficient drug development. So we need to know all these requirements. So if we get to work closer to FDA, to regulatory agencies who want to do that, take advantage of those meetings. Okay. Now, when we execute a clinical development plan, we want to plan well before executing, and want to make sure trials will support the label claim. And you look at different studies. Some studies you can develop them in a parallel fashion. Sometimes they have to be developed in a sequential fashion. And when you execute the clinical development plan, it's an iterative procedure. You're learning and you're improving, right? So you need to consider the non-clinical milestones when you're developing in the clinical development stage. You want to determine which objective will be answered by individual studies, which by pooled analysis. So we'll come back to this exploration and confirmation, all right? It's a phase 1, 2, 3, 4 development process. For exploratory, we do estimation, point estimate, interval estimate. For confirmatory purposes, we do hypothesis testing. It's a learning process of obtaining information and making decision. And design considerations for exploratory and confirmatory could be different. And analysis methods depending on objectives. For labeling, you may consider the entire database. Thank you. So I think with these 2 lectures, we completed this CDP, clinical development plan.